Good morning, everyone. We are Team Hemova. My name is Þóra Þorgísdóttir, and with me are Peter Lee, Shashira Nages, Mary O'Grady, and Sherry Hall. We work closely with three physicians from Johns Hopkins Hospital: Dr. Reifsnyder, head of vascular surgery at Bayview; Drs. Hong and Weiss, interventional radiologists from Johns Hopkins Hospital; and other clinical, business, and technical advisors. Together, we have developed a medical device that can potentially save tens of thousands of dialysis patients. This is Joe. He is a kidney failure patient who relies on dialysis to live. Worldwide, there are over 1.5 million Joes. Joe has a surgically created vascular access site in his arm that enables him to connect to the dialysis machine that filters his blood. These vascular access sites don't last very long and need revision surgeries costing Medicare over $1 billion every year. Joe has had 4 surgeries in the last 3 years and will soon run out of access sites. Without the ability to filter his blood through the dialysis machine, Joe will die. And this is a reality for tens of thousands of patients worldwide every year. We have developed Hemova, which is a solution to these patients, an implantable device that provides reliable vascular access. There are 2 main reasons why vascular access sites fail: infection and stenosis. Currently on the market, there are 3 main options: fistulas, grafts, and catheters. Shown on this graph in bubbles sized and representing their market share, and their placement on the graph represents how they effectively address these 2 different problems: infection and stenosis. In order to provide the dialysis machine with the high flow needed, grafts and fistulas connect arteries and vein in the arm, creating unnatural high flow in the vein. This unnatural high flow damages the vein and causes stenosis, or vein occlusion, resulting in an average lifespan of grafts of only 1 year and fistulas of only 2 to 3 years. Catheters, on the other hand, access high blood flow directly in the patient's heart. However, they have high risk of infection since they come through the patient's skin, resulting in an average lifespan of only 90 days. There have been several attempts in the last few years to address these problems. However, none of them appropriately address both the problem of infection and stenosis. We attended one of the biggest vascular conferences in the world in New York last fall, where 2,000 clinical specialists came together to discuss the problem of vascular access sites for dialysis patients. The conclusion was clear: there must be a better solution. We have developed the Hemova that addresses both the need for preventing infection and stenosis. So the idea came about last summer during rotations at the Hopkins Hospital. As we watched an angioplasty procedure where a fistula was being repaired, we learned about this clinical need. Initially, we focused on preventing stenosis. This can be done by accessing naturally high flow in the body, and with the help of our vascular surgeon, we've targeted the femoral vein as a good candidate for high flow with respect to safety and making the surgical procedure easy. Now we needed to get to the femoral vein in a safe way, because the femoral vein cannot be pricked directly for the dialysis procedure, and we wanted to do this subcutaneously, avoiding the risk of infection associated with catheters. The solution was a subcutaneous port system, a port sitting right under the patient's skin, connecting with tubing down to the femoral vein. Now, we wanted to minimize the artificial material in touch with the patient's bloodstream, and therefore developed a valve sitting right at the anastomosis site, or where the device is sutured to the vein. Our valve has been through several iterations with several different mechanical activation systems. Last but not least, we wanted to avoid our device from getting clotted and getting infected, and thereby we developed a system that enables you to flush out the whole device after each dialysis session by adding a 3rd tube and a center port. Here you see our device implanted in a patient's thigh, connected to the femoral vein, accessing naturally high flow, and here you see how the dialysis needles connect to the device. Now, unlike catheters, our device is completely subcutaneous, thus lowering the risk of infection, and unlike grafts and fistulas, we access naturally high flow, thus avoiding stenosis. Additionally, we have this cleaning mechanism that will prolong the lifespan of the device as well as the patient. Now here you see the device implanted again in the patient's thigh. With the valves closed, the blood flow of the patient flows freely past both valves undisturbed. Now when undergoing dialysis, the valves are opened up and the blood can flow through our device and into the dialysis machine and back into the patient. After the dialysis session, the valves are closed and the system can be cleaned out with a solution as seen in this picture. Now let's take a closer look at the valve design. Our valve is activated through pressure. So by pulling saline through the center port, as shown before, which results in pulling saline through this tube right here, the valve can be flipped up, allowing blood to flow into our device and undergo dialysis. After the dialysis session, the valve can be closed by pushing saline back into that center port and thereby flipping the valve down like this, enabling a flushing of the system without any interference with the patient's bloodstream. And you can see how the valve is designed to lay flush with the vein of the patient. Now this is our alpha prototype that we're currently using for initial functional verification through bench testing and short-term animal trials. We've already conducted 3 pig studies where we verified that our target vein has the flow rate and tolerates the flow rate needed for the dialysis machine, and the results were very positive. Now, before I hand this over to Peter, I want to leave you with the value of our product. Vascular access sites are extremely expensive for the healthcare system today, and even the most cost-effective solution, the fistula shown here, is not an option for a big portion of the patients. We've developed the Hemova that can be offered to all patients and will significantly lower these costs, and this is how. The Hemova is designed to last for 5 years, thus avoiding 4 interventional procedures at the cost of $17,000 per patient per year. This results in an annual cost saving for Medicare of $600 million. Or more importantly, I want to say, the Hemova can potentially save patients' lives and significantly improve patients' quality of life.
Thank you, Thora. So I'm going to talk a little bit about how we're going to get this device into patients. So first, a very broad level overview. Our target market is the 350,000 hemodialysis patients in the United States. To enter this market, we project to go through the 12,000 patients with no access options right now. These are the most desperate patients, and they need an access option to live. From that point, we will move on to patients without upper extremity options, and finally into the greater graft population, which we believe that our device will be eligible for. This market is growing by 100,000 new patients each year. It's also growing at a trend of 5.9% per year. This is driven primarily by the increasing trend in diabetes and hypertension, which are the leading causes of kidney failure. And this is not going to go away anytime soon. Our initial path to market looks something like this. Currently, we are constructing a prototype as well as bench and animal testing. We envision this to span the remainder of this year as well as 2012. At the end of that time, we will engage in a 6-patient pilot study, and we will also be looking for a management team to take us through the clinical trial stages, which we project to occur in 2013 for FDA clearance in early 2015 and market entrance shortly thereafter. In order to drive adoption of our device, we've identified several reimbursement codes already for DRG and CPT codes for the hospital and physician. These codes, we believe, can be used for our procedure, but we will also be applying to the American Medical Association for new codes that are more specific if necessary. In terms of regulatory strategy, we are a blood access device and are therefore currently classified under Class 3 of the FDA. However, after preliminary and informal meetings with Hogan Lovells and the FDA, we have reason to believe that we can go through a 510K pathway. This is because current blood access devices have not been called for PMAs, and until that changes, we believe that we can do 510K. What this means is that we can go through a 50-patient clinical trial to prove the safety and efficacy of our device. Our financial projections project break-even point in 2018 with around 5,000 units sold. We project net revenues of $40 million in 2019. The rapid increase in adoption, we believe, is driven by the 4-year patency trial, which we project to start in 2013 and conclude in 2017 to give physicians the evidence necessary to adopt our device into their patients for long-term and for the broader population of patients. In order to protect our technology, we filed for 2 provisional patents through the Johns Hopkins Technology Transfer Office and have analyzed patentability and freedom to operate. We've found really no other devices that access our unique method for accessing the blood vessels, the novel valve mechanism, which we believe has applications for other devices as well as the cleaning system. Our immediate goals for the next 12 months, I will be leading a team of engineers. I'll be staying here to continue development on this device, as well as working with our clinicians in vascular surgery and interventional radiology. And the immediate goals will be working to demonstrate 2 weeks without thrombogenesis, as well as ease of surgery and a beta prototype. Shortly afterwards, we aim to demonstrate 1-month patency, and our ultimate goal is to demonstrate long-term patency in multiple animal studies over the next year. These goals are driven by the talks that we've had with our investors that we've been talking to and a number of medical technology accelerator firms. They indicate that they would be highly interested in hearing about our technology as we develop it, as it is a very large market and would be able to address a very large clinical need. This includes a favorable exit option at this point in next year when we demonstrate a functional prototype with long-term animal data, as well as further down the line after clinical trial stages. We believe larger companies such as Covidia and Bard, they would be interested in such a technology. Our funding plan over the next year are to apply for technology accelerator funds from the CBID department as well as TEDCO from the state of Maryland. We believe that these funds will allow us to get through the next year of animal testing in order to bring us to the next stage of SBIR funding and further angel and venture funding to bring our product to market. Oops. This is just a brief list of some of the competitions that we've been to this year. We've placed very well at University of Oregon, the Rice Business Plan competition, and we hope to do so in the future as well. We'd just like to extend a big thank you to all of the clinical faculty, CBID faculty, and support staff, most notably Drs. Hong, Reif, Snyder, and Weiss, who we work with at the hospital, Drs. Charya, Ehrenheim, Yazdi, and Mary Beth and Sarah for all their help for us this year in getting our project to where we are. In conclusion, our vision is that we believe that no patient should have to die from a lack of dialysis access sites, and we invite you to join us later at our poster session, and we'll take any questions. Yes?
Let me just check if this works. I am Ching Xia, or is QX. Could you go back to the slide where you showed the device in the person's leg? Okay, so I was just wondering if you considered the surrounding anatomy of the leg, like the femur and the muscles, because this device looks kind of huge, and I was just wondering if it's possible for the person to walk or even run because of the interactions with the muscle around it.
Yeah, sure. So it looks probably a little bit larger in this picture than it actually is. The tubing are about 5 to 6 millimeters thick, and they go down to the femoral vein, and then they're tunneled up right under the patient's skin. There's room in a patient's thigh. It's fairly large, and you tunnel it in between the muscle up to the right under the skin. You see it's kind of supposed to be shaped in this picture, so it kind of wraps around a little bit. Maybe it doesn't show well enough.
All right, because so in this case, if a person runs or anything, would a muscular interactions like rip the device off the vein and cause a lot of bleeding?
Mm-hmm. That's a good question. We get that a lot. Actually, these patients are not very active. They're unfortunately very sick. So we're not talking about any type of running. We're just talking about walking around the most. But those are certainly items that we need to test further later down the line.
Yes, please.
Works. Almost congratulations. Definitely seems to be a very innovative design. So great work, team. My question is sort of related to the previous one, but what I'd like to know is a little more sharing of thought from your side on how did you were choosing the femoral as the access vein or artery, rather the artery, versus any other side.
Right. So this is something that we've been in a lot of talks with our physicians about, and so currently in catheters, they place a catheter directly into the heart to access a large amount of blood flow there. Unfortunately, that causes a large amount of the central venous stenosis and other factors that cause clotting within those catheters. So we needed an area with high enough blood flow, but that's also easy enough to access. We believe that the femoral vein provides this. The femoral vein has about 800 milliliters per minute of blood flow, and the dialysis machine requires about 400. There's a few other, and we didn't want to go into arteries because any failure within the arteries would be very detrimental to the patient, whereas the vein is not as bad. And so the femoral vein is really one of the best veins to do this in. There are a few other options that we are looking at, but right now we're just focusing on the femoral.
I think we can have further questions during the poster session. Thank you very much again. I'll ask